Hi Year 12s, we're working on the second video now for our cell division PowerPoint, which we're going to be looking at meiosis. So we've already looked at binary fission and mitosis, which are basically the cell division that occurs for somatic cells or for prokaryotic cells. We're now going to be looking at meiosis, which occurs only for gametes and produces our sex cells basically. Let's get started. So first off with the word I used before, gametes. So gametes are basically the sex cells in your body. And meiosis is the division of the nucleus that creates those gametes in your body. Meiosis results in 4 daughter cells, and each daughter cell has half the number of chromosomes found in the parent cell, so it's known as a haploid cell. Those daughter cells then differentiate to form gametes. Now male gametes are called sperm, female gametes are called eggs or ova. When a male and female gamete fuse in a process called fertilization, they result in a new cell with 2 sets of chromosomes. That new cell is then called a zygote, and it's the first cell of a new organism. That process needs to occur so meiosis itself needs to occur in order to produce genetic variability. We want genetic variability because species that have a high level of genetic variability have an evolutionary advantage, and their survival is increased. The chances of survival are increased. Now, we have to have the products of meiosis with only having half the number of chromosomes, or haploid, because when they fuse, you get that one cell that has the right number. So if a sperm has 23 chromosomes and an egg has 23 chromosomes, they fuse together to create 46 chromosomes, which is what the human needs to have. If you had a sperm with 46 and an egg with 46, then you end up with 92 chromosomes. Now we know that 47 chromosomes, so trisomy on chromosome 21, gives us Down syndrome. If you have 92 chromosomes, there's no chance that that offspring survives. So we need to go through meiosis in order to actually survive in general and have the right number of chromosomes in the zygote. Somatic cells. So somatic cells, we just need to look at this term before we start. Somatic cells are all the cells in the multicellular eukaryote that aren't the gametes. There's over 200 different somatic cells in the human body, and each type of somatic cell helps us in carrying out a specific life process. Somatic cells are diploid, which means they have 2 sets of chromosomes, whereas gametes are haploid, only having 1 set of chromosomes. The number of chromosomes in a diploid cell is referred to as the diploid number. So the diploid number in humans is 46, whereas our haploid number is 23. Meiosis. So in humans, there's 2 different types of cells that undergo meiosis. So we have spermatocytes and oocytes. Spermatocytes create sperm, oocytes create eggs. Meiosis occurs in 2 stages, meiosis 1 and meiosis 2. Let's look at the whole process of meiosis. So prior to the first stage, called prophase 1, DNA replication occurs, replicating the number of chromosomes that are going to be in the cell. So exactly the same as any other cell division. The first 2 steps here are the same as mitosis, so the chromosomes will start to condense and the nuclear membrane will break down. Then we'll have our centrioles move to opposite poles of the cell and begin to form spindle fibers. So both of those points exactly the same as what happens in mitosis in prophase. The new parts that are happening just in prophase 1 of meiosis is that homologous chromosomes, so chromosomes of the same type, so chromosome 1, chromosome 2, chromosome 3, they're the same type, chromosome 1, chromosome 1, chromosome 2, chromosome 2, they start to pair up. And they exchange DNA in a process called crossing over. So over here, let's say we have 2 chromosome 1s, they've lined up together, and they start to cross over. And they actually exchange genetic information. Now that could occur at a different point each time crossing over occurs, which gives us 4 chromatids that are genetically unique. So we end up with 1, 2, 3, 4 chromosomes, all of which are different. So it doesn't matter which sperm cell you get, or an egg cell you get, it's going to have a different chromosome. We then move on to metaphase, where the spindle fibers will attach to those homologous chromosomes and begin to contract. Those homologous chromosomes will then align in the middle of the cell. So we can see in this step here that they start to line up along the middle of the cell. The difference here between metaphase in mitosis and metaphase 1 in meiosis is last time we just had 1 chromosome lined up down the middle like this, whereas in metaphase 1 we end up with 2 lined up together in the middle. So in this diagram that I've got here on the right, it's only this first cell here that is metaphase 1. There is no order to the way that these homologous chromosomes line up. Okay, so you might have dad's or paternal chromosome 1 here, maternal chromosome 1 here, paternal chromosome 2 here, maternal chromosome 2 here. Or you might have paternal chromosome 1, maternal chromosome 1, maternal chromosome 2, paternal chromosome 2. So you see they've swapped around in that side. That's called random assortment. It doesn't matter where they've lined up. That just helps us to increase the genetic variability of the gametes. We then move on to anaphase 1, where one chromosome from each pair of the homologous chromosomes are physically pulled on by the contracting spindle fibers, and then you end up with one homologous chromosome of each pair moving to opposite sides of the cell, or the poles of the cell. The parent cell will then begin to elongate and begins its preparation for cytokinesis. We then go through telophase 1 and cytokinesis. So in telophase 1, the nuclear membranes start to assemble again, and the separated chromosomes are separated into their nucleus. Now it's worth noting this doesn't always occur. Okay, sometimes we're going straight into meiosis 2, and there's no point of recreating that nuclear membrane because in the first stage of prophase 2, it's going to be torn down again. Why waste energy building up to tear it down straight away? So it doesn't always happen, but it's worth writing that it does. Okay, just know biologically, doesn't always happen. Chromosomes will then also start to decondense and they'll no longer be visible. The parent cell, which was a diploid, then divides into a daughter cell through cytokinesis and becomes a haploid, and that's because it's only got one set of chromosomes now. Okay, you're physically separate. You had the two sets of chromosomes lined up down the middle, and we physically separated them. So it's at the end of meiosis 1 that your cells actually become haploid. Both daughter cells are haploid, as they contain 23 replicated chromosomes, as we've just talked about. We then move on to meiosis 2. So meiosis 2, we start with prophase 2, and the replicated chromosomes start to condense and again become visible under the optical microscope. And again, the nuclear membrane starts to break down and the centrosomes will move to opposite sides of the cell. We then move into metaphase 2, where the spindle fibers are going to attach to the chromosomes, which are now just one, and align them along the cell equator. So this is almost exactly identical to metaphase in mitosis, except for the step that these are now one of the homologous chromosomes. The spindle fibers continue to contract, and consequently they'll separate the sister chromatids, which are now called chromosomes. Those chromosomes move to opposite poles of the cell, and the parent cell again begins to elongate in preparation for cytokinesis. Last, we get to telophase 2 and cytokinesis. So the nuclear membranes assemble around the chromosome in each of the dividing cells, the chromosomes condense and are no longer visible with an optical microscope. The products of meiosis here are haploid cells containing a single set of chromosomes. Okay, so we've actually ended up, because meiosis 1 splits into 2 cells, and each of those cells then goes through meiosis 2, we end up with 4 daughter cells that are all haploid at the end. Now, this whole process wouldn't work unless DNA replication happened at the beginning. If DNA replication happened at the beginning, didn't happen at the beginning, after the first division, we'd only end up with 23 chromosomes all up, and they wouldn't be duplicated. So we wouldn't actually be able to end up with 4 haploid cells. So the reason that we have DNA replication take place in meiosis is that we can actually create 4 cells from each division. Just a little video of the meiosis summary that we'll watch here. So we start off with prophase 1. Actually, we might start off with DNA replication. Okay, so in our interface here, okay, we've then gone into prophase 1. So in prophase 1, we've had our DNA replications already taken place. We've got our centrioles on each side of the poles. Our homologous chromosomes are lining up, and crossing over is taking place. As we continue the video, we end up in metaphase 1, where the spindle fibers are contracting. We've got the homologous chromosomes going through random assortment, lining up. And as we continue along, the spindle fibers are going to contract, and they're going to pull apart the homologous pairs, which leads us into anaphase 1. We then end up into telophase 1, where our nuclear membranes are going to start to surround our new chromosomes, and we physically divide into 2 cells in cytokinesis. Okay, we're now moving on to meiosis 2. Okay, where prophase starts to occur. Again, our centrioles move to opposite ends, our spindle fibers start to extract, and they line up the individual chromosomes, or each one of the homologous pairs, along the equator of the cell. Now, again, we start to have the spindle fibers contract in anaphase, ripping those apart, and then we have our telophase, our nuclear membrane, forming around the now separated chromatids or chromosomes. And then finally those cells divide again, creating 4 different cells.